Our second session will start with Edward Grow, and he will be introduced by Laura Benazinski, Associate Professor in CISL and Ida Green Center for Reproductive Biology Sciences. Thank you.
Hi, everybody. It's really my pleasure to introduce the newest faculty member in the Green Center for Reproductive Biology Sciences, Dr. Edward Grow. Ed completed his graduate studies with Jana Wysocka at Stanford University, where he discovered that evolutionarily young endogenous retroelements are transcriptionally activated in human preimplantation embryos, demonstrating that development is compatible with expression of these retroviral transcripts. He then carried out his postdoctoral studies with Brad Cairns at the Huntsman Cancer Institute at the University of Utah, where he studied mechanisms regulating zygotic genome activation. He found that P53 facilitates the transcription of DUX, which is a key transcription factor required for zygotic genome activation. We were lucky to have Ed join the faculty of UT Southwestern in 2022. The goal of the Grow Lab is to understand how embryos navigate the egg to embryo transition during the embryonic genome activation. Ed's research program has 3 arms. First, he will understand the molecular determinants of embryonic genome activation across species. Second, to define the role of retroelement activation and silencing in this process. And third, to develop in vitro culture systems to produce high-quality eggs. These studies are fundamentally important for understanding the earliest stages of human development and will lay key groundwork for improving female fertility preservation strategies. I joined our faculty as the elusive molecular reproductive biologist that the center had been searching for over a decade. Ed's research program falls directly within the key mission of the center, using the most advanced techniques in epigenomics and gene regulation to answer long-standing questions of the utmost importance in early development. Ed's program further serves as a bridge to our clinical colleagues in the OBGYN department with real-world implications for improving human fertility and patient outcomes. So Ed is a true scholar, he's a generous colleague, and he's a real asset to this institution, and I hope you'll join me in giving him a very warm welcome to UT Southwestern.
Oh my gosh, Laura, thank you so much. That's very kind. So thank you again to the organizers for inviting me here today, and I'm excited to share our work. Today I'm going to tell you a specific story in our lab looking at oocyte identity during mouse in vitro follicle culture. So one of the holy grails of the reproduction community is to accomplish what we call in vitro gametogenesis, where we can produce sperm and eggs, the mature gametes, in order to treat infertile couples. This process entails the creation of pluripotent cells, pluripotent iPS cells from a patient, in which we can derive early germ cell fates. And in theory, we can produce gametes for people undergoing infertility. Although we can produce these early cell fates quite easily in culture, it's actually very difficult to produce these maturing gametes and the very end of the line, the sperm and the egg in vitro. And the reason why is because typically these cells develop in vivo in the context with somatic cells under the control of the endocrine system. And so recapitulating this process of gametogenesis ex vivo has been very challenging. In the Grow Lab, we're interested in female fertility, and this process involves the creation of follicles through folliculogenesis. So the follicle is really the functional unit of the ovary, and it encloses the oogonia in this structure that has somatic cells that grows, producing an oocyte that can undergo meiotic resumption and make an egg that can be fertilized. So this process is highly regulated in vivo, and in mouse model that we study, it takes around 21 days, but in humans, it takes much longer. So we're studying the mouse model in order to understand this process and the changes that happen during oogenesis and folliculogenesis. So an alternative to the process of in vitro gametogenesis is in vitro follicle culture. So instead of making oocytes or oogonia directly from stem cells, we can take the very early primitive follicles that exist in the ovary and culture them ex vivo in order to produce eggs. And so how this process works is we would take the oocytes that are enclosed in the follicle structure, embed them in a 3D alginate bead, and grow this follicle ex vivo to produce a large antral structure that contains the developing oocyte, which can then be ovulated to form an egg, fertilized to form a zygote, and then developing to form an embryo. So this process of in vitro folliculogenesis has been established in mouse, and it works, but it's quite inefficient. So we have problems in producing high quality eggs that can be fertilized or embryos that can undergo development. And so it's clear that during the process of in vitro follicle culture, there are changes that are imparted on the oocyte that impair its development after ovulation. And so we're really studying a key knowledge gap in the lab is how does this process of in vitro follicle culture alter the oocyte and its development? And so we expect that some follicles in culture are competent to mature normally, and these follicles would enclose high quality eggs, whereas many of the follicles that we include in culture deviate from that in vivo paradigm by some signaling pathway. And this is a really unclear molecular event that occurs in many of these follicles in culture. So our major focus is what is the identity of these oocytes from an in vitro follicle culture system. And so we use a technique, single cell RNA sequencing, in order to look at the oocyte transcriptome in a holistic way to understand how the process of in vitro follicle culture alters it. And so we are comparing the in vitro grown and the in vivo grown oocytes in this experiment, and we harvest the follicles when they reach their normal maturation stage. And so this is an example of one of these RNA-seq experiments shown on the right, where the starting material is a single oocyte right here that I'm showing you colored in pink. And the controls for this experiment are in vivo developed oocytes shown in green, or ovulated eggs, which are shown in brown. And I'm going to overlay the in vitro grown oocytes on this plot. So we actually discovered that there are two different identities of in vitro grown oocytes that we can recover in our system. There's a population of oocytes that we call class 1, and these oocytes are more similar to their in vivo controls compared to class 2 oocytes, which are shown in purple. And these oocytes are distinct molecularly from the population of class 1. So what is the difference between these two populations? It turns out that class 2 oocytes that we recover from our system, even though they're derived from antral follicles, which are morphologically normal, have a developmental delay in the oocyte compared to the class 1 follicles or the antral follicles from the in vivo controls. So what is specifically different between these two classes of in vitro grown oocytes? We're really interested in understanding whether they have different developmental trajectories, but of course, single cell RNA sequencing destroys the sample in order to measure it. And so we're currently identifying prospective markers, but in the meantime, we can perform some computational analyses that help us to infer the developmental potential of these oocytes. So we performed a computational analysis called RNA velocity that measures the intronic reads produced during transcription. And these have been shown to actually anticipate cell identity changes. They really anticipate what the cell will become in the future. And so this type of analysis is represented by a vector, which is a directionality and a magnitude. And we can see that the class 1 oocytes are developing normally, whereas the class 2 oocytes are confused about what they should become in the future. So what is the molecular difference between these two populations of oocytes that we've discovered in our system? We think that one of the key distinguishing features is the expression of oocyte-secreted genes GDF9 and BMP15. And you can see here that the class 2 oocytes have low expression in purple compared to their class 1 in vitro controls or the antral follicles, which are in vivo developed. And so this downregulation of this key oocyte secreted factors really jumped out at us as a key finding. So what are these factors? GDF9 stands for growth and differentiation factor 9, or BMP15 stands for bone morphogenetic protein 15. These are probably the most important thing that the oocyte makes and secretes into its follicle environment. So they're TGF-beta factors that are produced by the oocyte. They're secreted into the follicle, and they activate SMAD signaling in the cumulus cells. And that SMAD signaling helps to control the activity and development of both the follicle, and then it feeds back into the development and growth of the oocyte. And so we think that in class 1 oocytes, this signaling pathway is activating normally, whereas in class 2 oocytes, this pathway is downregulated, and there's likely impaired functionality and identity of the cumulus cells. So during oogenesis, there's a really delicate conversation that happens between the oocyte and the somatic cells, and that conversation is clearly breaking down in these class 2 follicles. So we're interested in whether the class 2 follicles have impaired cumulus cells and how that impairment of the cumulus cells affect the growth and development of the oocyte. And finally, we're interested in whether we can rescue that defect by supplying GDF9 and BMP15 to the culture media. That would be an easy fix. However, it's possible that there are more differences in the oocyte that distinguish them upstream of the expression of these key oocyte-secreted factors. And so if that's true, we're really studying the TF circuitry that controls the downregulation of GDF9, BMP15, these key oocyte-secreted genes. What are the transcription factors and what are their binding sites, either enhancers or promoters, regulatory elements that govern the expression of these key oocyte-secreted genes? And so with that, I'd just like to conclude I've showed you today that we've been able to molecularly characterize the in vitro follicle culture system for the first time, and we've discovered that they're actually cryptic heterogeneity in terms of the fate of the follicles in vitro. There's a class 1 oocyte that's very similar to the in vivo oocytes that we think would make a good egg, and there's a class 2 oocyte that we think is developmentally delayed. What we found was actually the deviation between these two oocyte fates might actually be caused by defects in the oocyte in terms of creating its own niche within the follicle environment by low expression of GDF9 and BMP15. So what are we doing in the future? We're really interested in determining whether the cumulus cell functionality is indeed impaired and how that affects the development of the oocyte. And furthermore, in the future, we're interested in whether changes that happen during in vitro folliculogenesis subtly alter the oocyte transcriptome or the epigenome, and that happens to impair the development of the embryo after fertilization. So with that, I'd like to thank the people in my lab that contributed to this work, including Chi-Chi and Abi, all of my colleagues in the Green Center, and our funding sources. And I'd be happy to take any questions.
Questions? Yes, we have Chi.
Really nice work, Ed. So just curious, what's your take on, how far away we are from complete oogenesis in vitro?
So you can make an oocyte with the mouse model, which is the most advanced, and many years have gone into making that system work, but the quality of the oocyte is quite impaired. So particularly in vitro, they don't enter into meiosis often. They don't extrude their polar body. So not only do they not undergo recombination, but they're aneuploid. And so I think that really the ultimate use of this is the in vitro folliculogenesis approach, because it takes the follicles that are already formed and enter into meiotic arrest in the ovary. And I think that that is what is going to fix this problem.
Another question here.
Hi, Ed. I wonder if you're looking at other species beyond mouse and human, because I imagine some of this has been done in livestock. And is that conserved in terms of the processes that are going on in those systems versus mouse and human?
Yeah, so I would have to say, if you look at the ovary across even vertebrates, typically, especially mammals, they all look essentially the same other than their different sizes. I would say that mouse is a great system because folliculogenesis is so fast, and so we can accomplish it really quickly. I think that that's what we're going to do first in the lab to really establish the molecular groundwork of how oogenesis occurs. But there are other large animal models that we would have to study in order to really apply this to humans, which is a very long time period of folliculogenesis.
Okay, we have a question here.
Ed, beautiful talk.
Thank you.
Thanks. Do you think that are those class 2? Is that class 2 population on a dead end branch of the pathway, or is it a population of cells that normally wouldn't accumulate and has gotten stuck?
So this is a great question, and we get it all the time from reproductive biologists. We don't know right now whether the fate of class 2 that we see in vitro is reminiscent of some fate that exists in vivo in the ovary, or if it's a completely different artifact of culturing them ex vivo. So I would point out, since you asked, I glossed over it, but there are some oocytes from the in vivo controls that cluster with this class 2. And we think that this is probably an early atretic follicle phenotype that we see. So in vivo, the follicles will either be ovulated, which is actually a very rare event, or they will die by atresia. And so we think that probably there's a hardwired program of follicle atresia that is set up that some of the follicles in vitro appear to take more frequently. Yep.
Okay, since we have a minute, I'm going to ask you a question. So mice and human are very different, right?
Yes.
And so the ultimate goal here is to make a human being. And I'm wondering what's your take on at what point you will be comfortable with the kind of thing you learn in mice and suggest it can be applied to human.
Right. So the very early work in this field, they've actually been able to do in vitro folliculogenesis in the mouse, make eggs, fertilize them, get blastocysts, implant them, and get pups. So it's clearly feasible in mammals to do, but I think that there will have to be a lot of work establishing this in a large animal model before anybody feels comfortable moving into other systems like human.
Okay, thank you. Thank you, Ed.